Okay, we are now moving on to 46B, where we're going to focus on the bulk flow, movement of mass amounts of food down a plant in the phloem. Again, it's not that diffusion and osmosis don't play a role, they do, but they are incorporated into a larger picture that we call bulk flow. Okay, so let's pick up here. Please remember we are talking about now phloem. We talked about how xylem works when we talked about transpiration. Now we're talking about phloem and the movement of of sugar from the leaves down to the roots traditionally. Okay, so moving food in a plant is called translocation. Okay, this term has popped up in used in different contexts. In this context, we're talking about moving food in a plant, large amounts of food, translocation from the leaves to the roots, moved by phloem. Sieve tube members are the actual phloem cells. They are lined up end to end, much like pipes would be connected from end to end. Okay. Phloem sap and xylem sap. Saps are liquids in a tree. Phloem is different from xylem sap because there's lots of sugar, because it's coming from the leaves where the sugar is made in photosynthesis, and we want to store that sugar somewhere. This is the concept you need to get in mind. Source to sink. The source of the sugar is, in our case, the leaves, and we're going to think of that sugar being transported down to the roots for storage. Understand that the source may not always be a leaf. It could be a photosynthetic part of a stem. It could be the dead of winter, and the source of our sugar is now going to be the root where we stored the sugar originally, and so on. But for simplicity's sake, the source for us in this discussion will be the leaf, and the sink will be the root, the place where we are going to store our sugar. Okay, so all right. Now, basically, the phloem is the plumbing. The sugar is made in a leaf. We have got to shove the sugar or load the sugar into the phloem. The phloem is going to allow the sugar to move down the plant, and then we're going to unload the phloem and stuff the sugar into a storage compartment, okay, in the root. So loading and unloading of phloem, shoving it in the phloem and taking the sugar out of the phloem. Okay, so sugar has to get loaded into the phloem cells, the sieve tube members. Remembering that sieve tube members are there to transport sugar, so it already has a lot of sugar in it if they're working. So we're going to have to use active transport to push even more sugar into a place where it's already high in sugar. So we're going to be going from low to high concentration. Remember, that is the definition of active transport. Okay? Now, we are going to move against the concentration gradient. This will require energy. Remember, our energy is always ATP. Okay? Now, the loading of sucrose into the phloem cells from the photosynthetic cells in the leaf where it's actually made is achieved by something called co-transport. We talked about this a long time ago. No doubt you've forgotten. I don't blame you. Okay? Essentially, we're going to use proton pumps. Okay? So here's a cell membrane, and that's a proton pump. And the proton pump, let's say this is inside and this is outside of the cell membrane. This proton pump is going to pump protons, let's say in this direction, so we get a bunch of protons out here, a high concentration of protons. Well, where would they like to go if they could? They'd like to go back to where there's a low concentration, but there's no door. We have to provide them with a door, a protein channel, if you will. Okay? However, this channel is going to have kind of a little catch in it. Okay? It's only going to allow the hydrogen ions to flow in if they are carrying with them a sucrose buddy. And so through this means of creating a concentration gradient for hydrogen, we will also allow sucrose to get pulled in against its concentration gradient. So the hydrogen ions flow with their gradient, and the gradient is so strong to pull them in that it will pull a coupled sucrose in. Oh man. That it will allow a coupled sucrose to come in along with it, okay, against its own concentration gradient, okay. Sucrose is going to be dragged against its concentration gradient because it is tied up to the hydrogen ion that's flowing in with its own gradient, okay. That is co-transport. Here's a little better picture than mine. What this shows is the high hydrogen ion concentration here, and we are going to allow that to drag a sucrose in with it through this means of co-transport. Okay? Now, again, the movement of sap down the phloem is greater than can be accounted for by diffusion, so again we have to address bulk flow. We're going to move things by pressure. Bulk flow, bulk flow is movement of large amounts of substances using pressure. Okay? In transpiration, it was a pulling or negative pressure. Here we're going to have that positive pressure. Okay, let me try to explain it. So this is representing the phloem. This is representing the xylem, and you notice they run parallel to each other. Where there's phloem, there's always xylem. Where there's xylem, there's always phloem. Now this is the photosynthetic cell that was creating the sucrose. Notice there's more sucrose here in the phloem than there is where it was actually created, because we're shoving it in there. So how can we get more in against its concentration gradient? Well, we would have this arrow here representing co-transport. So we're going to get this extra little sucrose in here. Now, please note, because we've got all this sucrose in there, we've got a high solute concentration and, relatively speaking, a low H2O concentration compared to the surrounding tissue. And notice there's a heck of a lot of H2O over here in xylem, because that's what it's carrying. Because there's so little water here, relatively speaking, compared to over here, water will flow by osmosis into the sieve tube member from the xylem. Now, we are packing this full of sugar and full of water, and when we pack this so full of water, we will create a positive pressure in the cell, and we are going to send this positive pressure wherever the pressure is lower so it can be released. Well, let's look down here. Here we have our sink. This was our source of sugar. This is our sink. And we are going to put sucrose away for storage in this root cell. Okay? As we do, we are going to take out these sucrose molecules. When we do that, we are increasing the water concentration is going up because we're taking, the solute out. The water then is going to flow from high to relatively low, because the mineral content when you are low in the xylem is a little bit more. So we're going to send the water back to the xylem, where it's going to be pulled up by transpiration again. Now, why did this water, this high pressure stuff, go down instead of up? Because we're relieving the pressure. We are relieving, it's not spelled right, I don't think, relieving the pressure down here, down low, so that if you squeeze the balloon, it's going to go to where the pressure is least. And so it's less down here because we're relieving pressure down here by removing sucrose and removing water. So the pressure pushes the sugar sap down the plant in the phloem, and that is translocation. We are moving large quantities of sugar and water by means of positive pressure. We're pushing it down the leaf, down the stem. Okay? So, the accumulation of lots of water in the top part of the phloem due to the accumulation of lots of sugar causes the pressure to increase. Pressure is relieved at the bottom. We've already talked about all this. Okay? This is called, you might want to know this term, the pressure flow model of translocation. So translocation and what we've just learned is the pressure flow model for how translocation of this sugar sap occurs. Okay? All right. So there we got it. We got transpiration. The cohesiveness of water is very important in pulling it up the stem, and the pressure flow hypothesis is critical in moving sugar sap down the stem in bulk flow amounts, large amounts, due to negative pressure over here, positive pressure over here. All right, hope you get that. If not, come to class with questions. Thanks for watching. I'll catch you next time.